Hi, everyone. Welcome to part two of regulation of gene expression. So today we're going to talk about the eukaryotes, our membrane-bound organelle kingdoms. Okay, so let's go through. Most of this, which is actually good for you, is that you already know how eukaryotes do some of the regulation of gene expression because of the RNA processing that takes place during transcription in the nucleus, as well as how codes are matched up during translation. That's another thing too. So you're going to get a good review of protein synthesis from chapter 17 as we go through this as well. Okay, so the first thing to note is that for eukaryotes, there's a decoupling of transcription and translation, which actually allows for more control points. Okay, so by decoupling, we mean that there is a separation through compartments. Eukaryotes have a nucleus with a nuclear membrane, and then translation takes place out in the cytoplasm. That's very different than for how it works with prokaryotes, where transcription and translation are coupled, right? So for the prokaryote, because there's no nucleus, transcription and translation happen at exactly the same time, and that is why prokaryotes have an operon. Several, actually. They have the inducible ones and the repressible operons, whereas eukaryotes do not have operons. That's something you want to keep in mind. We will do an FRQ about regulation of gene expression, and I do not want to see anybody talking about eukaryotic gene regulation through operons. That is not a thing. Okay? All right. So let's look at our eukaryotes. Let's go through this together. They regulate gene expression and therefore their metabolism, right? Metabolism is the sum of all chemical processes that take place in an organism to do their specific life processes. Okay, so eukaryotes regulate gene expression at every step of protein synthesis, from pre-transcription to post-translation. So we're going to go through those one at a time. Okay, and they are decoupled, and this is because eukaryotes have a nucleus with a nuclear membrane, and the mRNA is transported out of the nucleus. It carries the genetic code out of the nucleus because the DNA has to stay in here. Okay, translation taking place out in the cytoplasm. Oops, sorry. Yeah, that's coming for you. Let's go back. I feel like we're in class right now. Hang on. Here we are. Okay, so, oops. Yeah, I definitely feel like we're still in class, so have a laugh. Okay, so let's do levels of control, and let's look at what we've got inside this eukaryotic cell right here. Okay, here's your signal coming in. Here's your chromatin, right? That's the form that DNA takes inside the nucleus. And that chromatin modification, you can unpack that DNA involving histone acetylation and DNA methylation. So what does that mean? Well, the histones are the proteins that tightly coil the DNA into chromatin so that it can fit inside the nucleus. So acetyl groups can be added to that. Or we can remove some methyl groups, and it allows access to certain spots on the gene to make the gene available for transcription. So tightly packing or unpacking those histone proteins is going to allow access to the DNA so that genes can be transcribed. Okay, so let's take a closer look at that. All right, so here's the first level of control, and it's simply DNA access. You only keep necessary genes accessible. If you recall, we did an activity with transcription a couple of weeks ago where I asked the question within the activity, would a cell transcribe all genes at some point? And the answer to that is no, because number one, cells are specialized, right? So it's not necessary for you to transcribe every single gene inside a particular cell. And also keep in mind that your cells, your autosomes, right, that's everything except for the sex chromosomes, have exactly the same DNA, right? So it's all the same. So a white blood cell has the same DNA as a hair follicle cell. However, they're specialized, they have different jobs, and only certain genes need to be accessible for the cell to do that job, okay? So in eukaryotes, DNA is wound around the histone proteins. Let's take a look at what they are, right? So here's your histone proteins. You can see the blue is actually the DNA, right? And the proteins wrap this and tightly coil it up. Okay, so if you take the addition of acetyl groups, right? Here's the acetyl group. And you add those acetyl groups to the histone proteins like this, it unwinds and exposes sections of the gene. And then those genes can be transcribed. Okay, so if it's in this form over here, that's your heterochromatin, more tightly packed. This is unavailable for transcription. In euchromatin, that's right here, it's less tightly packed DNA. That would be available for transcription. Okay, so this is one way that you can actually regulate the expression of genes or the proteins that are produced, right? Because if we take off the acetyl groups, boom, you're right back here, right? Unacetylated histones. And the DNA would be tightly packed and it would not be available for transcription. Okay, versus our euchromatin over here that is available. Okay, so we can either add acetyl groups, that opens up the DNA for transcription. We can actually methylate the DNA, where we do the same thing with methyl groups, and it would also allow, if we demethylate the DNA, would allow for transcription as well. Okay, so cool fact, histone acetylation patterns seem to be heritable. You inherit some of those patterns from your parents. That's called epigenetics. So we say epigenetics strikes again. I'm going to do a whole lesson on that. Epigenetics is pretty cool. Okay, so recent research shows that even in interphase, right, the cell doing its normal thing, G1, S, G2, the loose chromosomes occupy distinct nuclear regions. Okay, so they're found in a distinct portion of the nucleus. And transcription factories, these are areas of the nucleus where active regions of different chromosomes can interface. And so that may be associated with certain common functions. There are thousands of transcription factories in any nucleus. All right, our second thing, we're still in the nucleus now, right? Because this is pre-transcription, and in a eukaryote, transcription takes place inside the nucleus. Okay, so all actors need to be present for the play to begin. So we're going to go through some of the things that eukaryotes have that prokaryotes do not, and take a look at how it affects transcription, right? So it's another form of regulation of gene expression. Okay, so eukaryotic genes interact with many upstream regulatory elements. Okay, so if we look at the model here, upstream, here's upstream right here. Here are your exons, here are your introns, and we can see the introns get cut out in the mRNA processing. This is considered downstream. So right here, these are the genes in the blue to be transcribed. Okay, so upstream of those genes, you can see these proximal control elements. They're pictured in yellow. You've got some enhancers, or what we call activators in eukaryotes. Okay, they're DNA sequences that precede a transcription unit that need to have specific proteins present for RNA polymerase to actually begin transcription. What does that mean? They're transcription factors, okay? So these activators allow for transcription factors to bind to the promoter section so that RNA polymerase can then transcribe the genes downstream. Okay, so if you recall, eukaryotes must have transcription factors present in order for RNA polymerase to bind to that promoter, boom, right there, so that it can then transcribe the genes. Okay, so that's part of that pre-transcription control. Okay, so the proteins that Mediate this are transcription factors, okay? So let's go through this model. Here's your activators, right? It's an enhancer region on the gene, just gene sequence. The activators will bind here. Here's your promoter, your ta-ta box, and this is the gene to be transcribed. Okay, so if we look at what happens, here's your activators. This kind of loops up, bends around. Here's your mediator proteins, your transcription factors are over here. Okay, so this whole complex is required in order for eukaryotes to have RNA polymerase to then bind to the promoter, transcription initiation complex, and then RNA polymerase is allowed to transcribe the genes. Okay, so what's our measure of control here? A few things. Okay, if the activators are not present, transcription cannot take place. If transcription factors are not present with the mediator proteins, RNA polymerase cannot bind and transcription cannot take place. So there's a few measures of control here for eukaryotes. Okay, so control of the transcription factor availability is one of the major ways that cells of a multicellular organism accomplish this differential gene expression, which in turn allows cells to differentiate to serve the different functions in an organism. This is so crucial for eukaryotes. It explains the level of organization, right? You have your cells, which form tissues, which form organs, which form organ systems. All of this is specialized, yet all of your cells, except for your sex cells, contain the same exact DNA, right? So that's in this model, we can see what's going on here. Okay, so if we look here, here's the enhancer sections of the DNA, here's the promoters, right? Here's a liver cell nucleus. Here's a lens cell in your eyeball, okay? And if we look, there are available activators for the liver cell to express the gene for albumin. That's protein, okay? And a certain type of protein, actually. Okay, and then that's going to be transcribed so that that gene can be expressed in a liver cell, right? But if we look at your eyeball is super specialized for vision. So there are available activators here, and if we look at the activators. Do you notice how they are different than the activators in the liver cell? And that should be, because each cell is specialized. Okay, so here, even though the gene is there, the gene for albumin exists in your eyeball cell because the DNA is the same. However, it is not going to be expressed, okay, because this is not a liver cell, right? And the activators to allow albumin to be expressed are not here. Okay, so we can see here's the crystalline. That's the gene to be expressed in your eye, and so that's activated for the lens cell, but not the liver cell. Okay, so that's what we mean by differential gene expression. Crucial for eukaryotes. Okay, number 3 is post-transcription. So that means lots of options lead to many different outcomes. And so we're going to revisit our RNA processing here, and I'm going to explain how alternative splicing of the exon actually allows for gene products that are different to be made from a single primary transcript. Okay, so let's take a look at that RNA processing again. Here we are, right? Here's your section of the DNA upstream of the gene, right? Distal control elements. Here's your proximal control, it means closer to the promoter. This is where the activators will bind to. Exon, intron, right? And so we all remember that the spliceosomes are going to cut out these introns, and then your mRNA has been processed. You've got your 5' cap and your poly A tail, okay? So like, here's your RNA processing in a nutshell, right? Following transcription, 5' capping, 3' poly A tails are necessary for eukaryotic mRNA to remain functional and then be transported out of the nucleus and into the cytoplasm, right? So that's the crux of RNA processing that we covered in class a couple weeks ago. All right, so what's unique for eukaryotes is that you can actually alternatively splice out some of the exons, and this allows for several different gene expressions for a particular cell. Okay, so anywhere from 75 to 100% of human genes with multiple exons undergo alternative splicing. Okay, so shown here, you've got a troponin T gene. which produces 2 different mRNA sequences for 2 different gene products. Not shown a Drosophila, it's a fruit fly gene, has enough exons to produce 19,000 different transcripts, which is pretty cool. Okay, so here's our troponin T gene. Here's your exons, right? So you can see them numbered right here. Here's the primary transcript, and we can still see those numbered pieces here. The lighter pink represents your introns that must be spliced out. So with alternative RNA splicing, you just look at the different combinations. Okay, so if here we splice out the 4, you can see the 4 is missing here, and you've got genes 1, 2, 3, and 5. This is going to express a completely different protein than if you splice out the 3 and you end up with 1, 2, 4, and 5. And the reason for that is the code is different here in the transcript, right? And so if the mRNA code is different for 1, 2, 4, and 5, then when translation occurs outside in the cytoplasm, there will be a different amino acids brought over here to this section of gene, and therefore a different protein will be made, right? So the genes are expressed differently. So that's alternative RNA splicing. Okay, number 4, pre-translation. Okay, so this is before any type of translation of a protein can take place. This is, ribosomes blocking the ribosome from actually translating the mRNA tRNA complex. Okay, so there's still lots left to learn. Recent research has uncovered many forms of what we call non-coding RNA molecules in cells. So there's a huge list. You can go to Wikipedia. I mean, this is as of October 2011, so I'm sure it's been added to since then. There are tons of different types of RNAs. Okay, we're going to look at one example, active in regulation of gene expression for post-transcription and pre-translation. Oops, typo. Okay, so here's your RNAs. Let's pop over here. Okay, and the one we really want to focus on is RNA interference, okay, mediated by a group of tiny RNA molecules. We call them microRNAs. The microRNAs are produced after the transcript for them is cleaved into multiple fragments by a dicer protein. Okay, so basically, RNAi acts as a cop, in quotes. Do not, if you have to explain how RNAi works, I'm going to give you an analogy, right? But this is what, you cannot use an analogy in an FRQ. This is only to help you understand, okay? All right, so this won a Nobel Prize in 2006, and I'm going to show you the little video of how that took place, okay? But basically, here's what we've got. We've got your primary microRNA transcript. It has a little dicer protein here. So these RNAis can chop up the transcript. It's part of the reason that the 5' cap and the poly A tail are needed, because the RNAis are present, and if they chop up the mRNA and degrade it, then of course translation is going to be blocked. Okay, so let's take a look at how that works. At some point, and I'm waiting for the YouTube, and here we go.
Every creature is made from a recipe that comes from its DNA, spelled out in chemicals A's and C's and T's and G's inside the famous double helix. So how do you get from DNA to become a real creature? Well, let's take one of those fantastic voyages and we'll show you. We're going to find DNA and, well, we'll make it a typical cell, so we're going to have to fly in and then go off to the nucleus of the cell, which will make a beautiful castle, the headquarters. And there's the DNA, the master code, inside the nucleus. DNA never leaves the nucleus.
You ever meet one of those mean librarians,
Yes.
special reserve section?
The one that go, pow.
Right, you can take the thing, you can copy it, but you can't take the book, because somebody else might need it.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that scribe, copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm C that makes up most of the cell, all those recipes floating through the air, they are RNA. And to finish up, in that C, you see hundreds of thousands, well, we've made them into little guys with chef hats.
Those would be ribosomes, and in your world, there are chefs who are using the recipes that are written in the RNA.
And whenever a recipe lands on a chef, whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. Proteins build cells, bone cells, brain cells, all cells. So all these chefs are basically building you. You are made of proteins. And the discovery of RNAi was an accident. It was a puzzle that appeared in a petunia. Maybe this. In 1986, geneticist Rich Jorgensen was working at a biotech startup company in California. He was asked to create a spectacularly dazzling flower. And so they decided to create a very, very, very purple petunia. Rich knew which gene produced purple. He knew how to sneak an extra copy of that gene into the plant's DNA, the master text, to be copied by that monk-like scribe.
It will be transcribed by the monk the same as any other gene. And he'll throw the transcript out the window into the cytoplasm, where the chef will be able to pick it up and use it.
Rich thought that if he added more purple recipes, he'd get a purpler petunia. So he did it, and he waited. And what happened?
We produced, instead, white flowers.
White flowers?
Complete opposite of what we had expected. Our initial reaction was that something must have been wrong with the gene that we had engineered to introduce to the plant.
A mistake?
A mistake. So we checked everything out, and there were no mistakes that we could find.
The petunia was a big puzzle. Nobody understood why, when you add an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
Quite by accident, Rich had discovered a secret inside living cells. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks.
The sole purpose of that virus is to make additional copies of itself, to the point that the entire cell is filled up with this. And the cell explodes, releasing these viruses to go and then infect whatever other cells they can find.
So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape. So when they see it in that shape,
They say virus. They say,
uh-oh. And the cop destroys the recipe.
cells were able to tell that something was very funny when they saw mirror image messages and start not just destroying the messages, but destroying anything that looked like that message. They'd worked out this whole defense system against viral RNA, and we then accidentally stumbled into using it.
The question, remember, was when Rich tried to make his petunia more purple, why did it turn white? Well, the answer, it turns out, was that Rich, by accident, discovered the cop. When Rich invaded the petunia cell and inserted his make more purple instructions, he didn't know it, but his purple instructions happened to have that suspicious viral shape. So when the cop saw the recipe, the cop thought, virus, and destroyed every recipe for purple in the cell. If there are no recipes for purple, the chefs don't cook purple.
And because there's no purple pigment produced, the flowers will be white.
And that's how Rich and his petunias helped discover what we now call RNAi.
RNAi means RNA interference, because the cop is interfering with RNA messages, with the recipes in the cell.
All right, so keep in mind, right, when we go to describe things, here's your chef. They're talking about the ribosome. DNA is locked inside the nucleus. RNA polymerase is providing the mRNA transcript that has to leave. And that transcript, if it doesn't look right, can be chopped up by RNAi or the cop. Okay, however, please, when we go to write about this, when we write about FRQs, Don't use the analogies of the cop. Don't talk about the chef. Okay, let's use actual terms and structures of the cell. Okay, so our last and final one is going to be post-translation. Okay, so this means that the ribosomes interpreted the mRNA, tRNA codes, the protein was formed, so post-translation is after that process has taken place. Okay, and in cells, they don't let unnecessary proteins hang around. Okay, so we've got ubiquitin. It's a protein so plentiful in all eukaryotic cells. It's called ubiquitous. And it will tag unnecessary proteins for transport to a proteasome. So if you recall back in the beginning of the year, we learned about proteasomes inside eukaryotic cells. They help to break down certain proteins. They are particularly abundant in eukaryotic cells, especially in your liver cells, in order to break down things that are toxic for you so that you can perform your life functions. Okay, so here's your protein to be degraded. Here's ubiquitin pops in. And once it's working on that ubiquitin protein here, it can be recycled, and so you can see the protein fragments that actually come out, and this little structure here is the proteasome that's taking care of that, right? So you can see how it's just chopping up the protein so that the gene is not actually expressed. Okay, so that would be the fifth way that eukaryotes regulate their gene expression. Ta-da! Okay, so that's all of eukaryotic gene expression. I don't think it's that difficult. If you have to recall or describe certain things, you can always talk about alternative RNA splicing because you should have a good understanding of mRNA processing in the eukaryotic nucleus. You can talk about pre-translation and RNAi. And I will also post a link of this Prezi so that you can go back through it yourself on your own time if you want to watch the video again, because I think it's super, super helpful. All right, so I will do a different video where we'll use my typical whiteboard setup. I'm super excited to do that, where I write on the board, we'll compare out and contrast the prokaryotic operons to eukaryotes. I'll do a different lesson going over inducible and repressible operons as well. Things that you need to know. Make sure you can explain the structure and function of all expression control systems in prokaryotes and eukaryotes. So you should be able to describe for prokaryotes inducible and repressible operons and the differences between them. For eukaryotes, you have to be able to explain the 5 points of control for regulation of gene expression that we just went over in this lesson. You're going to compare and contrast the expression control systems utilized in those cells. You will relate those control systems to how the cell is organized, right? So for prokaryotes, transcription and translation are coupled. Feedback mechanisms, so looking at, the operon is doing negative feedback mechanisms based on what's going on in the environment around that single cell. Okay, for eukaryotes though, the control systems are different. They are decoupled that we just went over. All right, so I hope that's helpful. And until next time, study well. Miss you guys.